All right, so on to gene expression. So I'm going to give the first part of the talk. I'm going to talk about the data we've generated, and then Joan's going to talk about how she's been manipulating and analyzing the data using bioinformatics and trying to get a systems biology approach analysis of it. So the four of us who are involved in this project are Helen, who's at the back there. Wave at us. Thank you, Helen. She does all the work. All the important work, and then there's Ken Jones over here on the left in the yellow shirt, and me and Joan are going to talk in a few minutes. So we had three original aims. I'm going to talk about the first two aims today. One is looking at the transcriptional dynamics of the face, so what genes are expressed in the face, both large RNAs and small RNAs. And then later on, we've begun the analysis of looking at bioinformatic analysis of differential splicing and differential isoform usage in the developing face, which we'll show is really important. And the last part, aim 3, we haven't got onto yet. That's not part of our milestones until the end of next year. We've seen already this morning several talks using looking at the developing mouse face. And thank you, Chris. And the developing mouse face really develops very quickly over time. This would be the first trimester in a human. And so you have these blobs here, which get filled up with neural crest cells, which we've heard about migrating into them. And they undergo morphogenesis and they undergo fusion, so that over like 3 days, you've got these distinct prominences which have turned into a mature platform, which is all fused together. And if you look at the cells in these prominences, they look just like round amorphous cells. They're mesenchymal cells, and they all look the same. And yet those different prominences are going to give rise to different components and different tissues. So we want to know what's driving those types of decisions in the developing face. Okay, so we've heard a lot about the neural crest. So a lot of your face is derived, or pretty much all of it is derived from the neural crest, which is going to migrate down from the neural tube into the developing branchial arches and frontal nasal prominences. And if you section through a mouse head, and here's using Rosa 26 reporter from Phil Soriano, either with Wnt1 Cre or an ectodermal Cre recombinase that we made, you can see the neural crest contribution here in blue and the ectodermal contribution in white, or vice versa down here. And so your neural crest are going to form, along with the mesoderm, are going to form the building blocks of your face, the skeleton, the muscles, whereas the surrounding tissues, especially the ectoderm, are going to provide signals for growth, and they're going to be part of patterning and morphogenesis, and they're also going to form things like your teeth and hairs and glands. But what I want you to notice from this is, so a lot of the stuff we've heard about already about gene expression, is that most people often take the mandible, for example. But the mandible is composed of both ectoderm and mesenchyme, but there's a lot more mesenchyme around than there is ectoderm. So if you do any gene expression studies, you'll often find that the ectoderm is underrepresented. And I wanted to say that both tissues are very important for development. Here's the same gene. So this is a transcription factor, which is presumably driving targets in either the ectoderm or the mesenchyme. And this is a mesenchymal knockout of 2 AP2 transcription factors. This is work from Eric in my lab, who's also somewhere here. Here's a control mouse, and here's this mutant mouse where you knock out these AP2 genes in the ectoderm. In that mesenchyme neural crest, you get this midfacial clefting here. But if you knock out the same genes in the ectoderm, you end up with this very different craniofacial phenotype. So clearly there's different programs that these genes are regulating in the ectoderm versus the mesenchyme to regulate craniofacial development. All right. So the approach we use then to try and understand gene expression at these stages before the face is fused into the final platform is we take mice at various stages of development, 10.5, 11.5, and 12.5. Before 10.5, there's not really enough tissue from the various prominences to dissect. After 12.5, the individual prominences are not distinct anymore, so we can't isolate them and analyze them. We dissect those out as the various prominences, then we treat them with dispase to make either ectodermal components or mesenchymal components. Here's Helen's dissections down here with the intact face, and then she can take off the ectoderm and make an ectodermal RNA component or a mesenchymal RNA component. And then we can look at the RNA. For example, we can use messenger RNA for RNA sequencing. And for these time points and these prominences and these tissues, we've now completed that analysis, and it's almost ready to go off to the hub. And this is the PCA analysis, the principal components analysis from that data. And the first thing to notice is that the replicates are really good. So it's very reproducible data, even though we're doing these microdissections. Ectodermal derivatives are shown in these diamonds, and mesenchyme with these triangles, and then the various FMP, MXP, or mandible are shown with these colors. And what you can see along principal component 1 here is that you're separating basically according to whether you're mesenchyme or ectoderm. So the biggest difference is between those two tissues in gene expression. We also dissect out the nasal pit separately because that's going to give rise to a lot of the olfactory neurons, and they have a very different program. And if we mix them in with the ectoderm or the mesenchyme, it really messes up our analysis. So we have a separate analysis of these components as well. And then you can see the next principal component separates according to time, 10.5, 11.5, and 12.5. And to a lesser extent, not even in principal component 3, we can see differences between the FNP, the MXP, and the mandible. So those are not the biggest differences out there. All right, so the data looks really good. And in the second part, Joan's going to tell you what we can do with this kind of data. The next thing we wanted to look at, so we looked at the large RNA population, the messenger RNAs. We also know from various studies that microRNAs are very important for craniofacial development. And there's increasing evidence that ribosomopathies are important for craniofacial development as well. So we wanted to have an idea of what other small RNAs, things involved in splicing, things involved in RNA pol-1 transcription, are doing during facial development. However, many of these small RNAs are not polyadenylated and they're differentially modified, so they're not that easy to do using RNA sequencing. So you could analyze them by sequencing, or you could analyze them by microarray analysis. And there are advantages and disadvantages to both, but eventually we came down on the size of using a microRNA for a bunch of technical reasons, including the ability to detect a broader range of RNA classes like tRNAs, as well as their reproducibility compared to the very involved protocols of looking at microRNAs by RNA-seq. Now, the disadvantage of using a microRNA is that you can only put on stuff there. The only stuff you're looking at is stuff you've put on the array, so you're not going to pull up anything you don't know about. We found there weren't really any suitable mouse-specific microarrays out there for looking at small RNAs, so we worked with AFI to design our own custom array. And on that array, we put on all the mouse microRNAs we could find, along with tRNAs, mitochondrial tRNAs, SNO-RNAs, SNRNAs, any other good small RNA we could think of. And then we also contacted Axel with his spoke to look at the possibility of studying enhancer RNAs, which are really uncharacterized or fairly uncharacterized class of RNAs, and we hope that we could interrogate those as well. So enhancer RNAs, which I don't think I've talked about today, are RNAs that seem to be transcribed from the vicinity of those enhancers that Axel was telling you about, which are expressed in the craniofacial complex, but probably also with other enhancers in other positions, and they help define whether an enhancer might be active in a particular tissue or not. So we just got the results from these RNAs, from these small RNA microRNAs last week, and we didn't originally want to use so these are brand new, they've never been used before. We didn't want to use our precious samples that we have for FaceBase on it right away in case we blew them. So we instead used triplicate samples from E16.5 secondary pallet, ectoderm, for a test run. And the good news is we can detect miRNAs, tRNAs, SNO-RNAs, SNRNAs, and 5sRNA very well. The data are really reproducible, so that's good as well. And we can detect the sort of microRNAs you might expect to be expressed in the skin, on the palate, including these ones here. And we also happen to have some Dicer knockout embryos around that hit the palate. So down here is a volcano plot from Helen. The stuff that's in red is stuff that's going up in the Dicer mutants. And the stuff that's in green is stuff that's going down in the Dicer mutants. And this is a log 2 scale over here. So you can see most of the stuff that's going down by a lot is in green, and all those things, for the most part, or actually all of them, are microRNAs. So that looks really good, and now we're ready to go ahead and use those arrays for the samples that we have already for the various prominences, for tissue, and for time. So that's good. We didn't see a lot of eRNA expression, and there might be various reasons for that. The wrong tissue, for example, or the wrong amount of RNA, or the wrong size of RNA, so we need to troubleshoot the eRNAs further for the future. Moving on to AIM2, we are now ready, because the data looks so good, to go and look at differential isoform analysis. I did some database mining to look at what might be differentially expressed. And these are examples that represent differences between the ectoderm and the mesenchyme for splicing. And here's screenshots of 3 genes. This one, S100A16, which is a calcium channel, has differential promoter usage between the mesenchyme and the ectoderm. Left 1 has a differential inclusion of an exon, and FGFR2, we heard from Phil Soriano earlier, has differential splicing between the ectoderm and the mesenchyme to produce different receptors. So these are functionally significant examples, and I think there's many more in the data set. So we're ready for this, for a more in-depth bioinformatic analysis there. All right. So our preliminary conclusions from the differential isoform analysis is most seems to occur between the different tissue layers, and many of these correspond to genes associated with EMT in other biological contexts. So what I forgot to say earlier is that Russ Carstens knocked out a gene called ESRP1, epithelial splicing regulatory protein 1, one of the functions of that gene is to differentially splice the FGF2 receptor between ectodermal and mesenchymal forms. When you lose that epithelial-specific splicing factor, which theoretically would destroy a lot of epithelial-specific splicing, you get bilateral craniofacial clefting in the mouse. So that's why we're interested in this isoform analysis, because we think there are a lot of genes in there which are required for face development. And a lot of those genes we found so far seem to be associated with EMT in other biological contexts. We don't see a lot of isoform differences in common transcripts between the 3 prominences, and there might be some alterations in exon usage over time, but that's not clear yet. We need to do some more analysis on that. So our plans are now to apply detailed bioinformatic and experimental analysis to verify those isoform differences using transcriptome arrays, et cetera, et cetera. So I'm going to stop there. That's the sort of straightforward data we can do a data dump with, but the data itself is not particularly useful until you can analyze it in some meaningful way, and Joan's going to talk about that more now.
So for the next 10 minutes or so, I want to talk about so we have a lot of data. We have a ton of data. Small RNAs, large RNAs, and there is also a ton of data at FaceBase and at Geo that's complementary. Yang's data with maxilla and mandible, proximal and distal. There's data out there from tooth and tongue and sensory, olfactory epithelia. So there's a lot of face-relevant data out there. And really the question is now, how can we integrate this and represent it so that it is helps us to understand the processes that are going on and becomes useful to many people for many different kinds of questions. There's Yang was talking this morning about these beautiful matrices that lay out things in terms of anatomy and age and are really organized in terms of what gene is expressed when and where. Okay, that's a tremendously important thing. And we have the anatomy that Shanna was talking about and that Yang was talking about that really maps in a macro scale the gene expression onto the tissue morphology. So that's very useful. We also have at a very micro scale the sorts of gene regulatory networks that Eric Davidson has pioneered that really express the transcriptional regulation and the cascades that underlie a lot of developmental processes we're looking at. And there really seems to be something missing in the mesoscale describing the cellular processes. Okay, so that's just setting up where I'm coming from as a cell and developmental biologist and thinking about how we might be able to use this data and to illustrate all these different scales. So just working off of the data set that we have that Trevor's described, where we have, let's see if this is going to work. Yes, okay. Radical differences between the gene expression programs in the ectoderm and the mesenchyme. And as he alluded to, we actually know quite a bit about the mesenchyme programs and much less about the ectoderm. So I want to just focus for a few minutes on what's going on in the ectoderm and use that to begin to illustrate these ideas. So about 1,200 genes that we've identified as ectoderm enriched by these criteria, and about that many in the mesenchyme. Of those, 3/4 are really expressed across the ectoderm all the time, and then there's a smaller group that's either prominence or age-specific, more dynamically expressed. And so beginning to dig into this, what's going on in the constitutive ectoderm group, you do the sort of normal things. You throw your gene list at David or GSCA or one of these other things and ask what functions, what annotations, what pathways. And what we see is what we would expect. We see a lot of genes involved in epithelial morphogenesis, the various annotations that are associated with that. A lot of things, another 100 or so, associated with the sort of cell biology structure, cellular structure that's associated with epithelia. Some odds and ends about growth and proliferation, and the very largest group associated with signaling. So this is to emphasize the really fundamental importance of signaling in the ectoderm and from the ectoderm to the mesenchyme, as Phil was talking about, in facial development. Well, this is a very broad, soft focus kind of view, but when you're talking about 850 genes, it's about all you can do. But to get some more insight or more detail, looking at the group of genes that are now dynamically expressed in the ectoderm, either prominence specific or age specific, and we can begin to get a little bit more insight there. So to deal with this, we have about 350 genes that are dynamically expressed. And so first, to group them according to when and where they're expressed, what the dynamics are. So with some hierarchical clustering, we come up with 13 sets of genes, which we can then interpret according to when and where and what's inside of them. So for instance, this first group here is really highly expressed in the 10-5 sample from the mandible. And when we look at those 37 genes and what functions are associated with them, we see, again, a lot of genes involved in signaling, a little bit involved in cell cycle, as you would expect for an early growing tissue, a little bit in cell shape, a little bit about cell energetics. And as we look across the rest of these genes, as we get older, we see the predominance of cell shape and signaling transitioning to a predominance of things having to do with epidermal structure and more specialized functions like secretion, lysosomes, stress responses. So this is all very nice. It makes sense. And then if we go on and look at what's prominence specific, we start to see some more fun things. If we look at what's specific to the FNP, the nose that contains the olfactory tissue, we see placodal genes. And as you go older, you start to see a lot of neural functions. And then in the late FNP, surprisingly, preponderance of genes having to do with melanocytes. Stop and think for a minute. Okay, this is where the vibrissae are forming. This is where the melanocytes are invaded. And so it's something that's kind of unique, and it makes sense in this context. If you look at what's happening in the mandible, we see things having to do with oral epithelia and dental placodes, and again, oral epithelia and secretory type functions. And so these make sense. again, in terms of the functions that we expect in these tissues and times and places, what's going on from the ectoderm. But again, this is not a terribly, or I think there's probably a better way of looking at the data. And so now this is what I wanted to put up as a sort of a cellular model, sort of a more mesoscale representation of the ectodermal program at E10.5. And so what this is meant to represent is a couple of epithelial cells adjacent to each other with their apical and basal surfaces and their tight junctions, their nuclei, their secretory apparatus, and the 50 genes that are in the early and E10.5, E11.5 mapped onto this. So we have a little bit of cell cycle, we have a little bit of things involved in cell structure, a little bit of things involved in interaction with the extracellular matrix. We have some things involved in cellular homeostasis and energetics, and a small but significant number of things involved in the other chromatin structure, licensing, new gene programs, some significant transcription factors, a whole mess of things involved in signaling. And when it's laid out in this way, a couple of things really stand out to me, along with everything we expected. There's this peculiar set, just two enzymes, but involving generation of catecholamines, very specific to this early ectoderm, raising the question of what is the role for catecholamines here, and something we haven't really thought about very much. Now, I could go on for an hour describing what's happening in every tissue at every stage, but this is, I think, to me, perhaps, a useful representation for these genetic programs. And I'm very interested in feedback from all of you. Is this useful? How could it be more useful? What would be improvements in this? All right, so. Okay, I've run out of time, and so no, I'm not close to being finished, but I will very quickly run through the second piece of this now, which is another way of thinking about these things. And this is based on the notion that genes that share functions are going to be co-expressed. And so a way of arranging gene expression data would be to sort all the genes into co-expression modules and then ask what's going on in each group, and is this a useful way of looking at the data? And so since I've run out of time, yes, we've generated a bunch of co-expression modules, thought perhaps to focus on the ones that are enriched in genes that have to do with clefting, because we understand clefting pretty well, better than some things. There's a dozen of those. And to really look at them and ask, first of all, are these co-expression modules, do they really have functional links inside of them? Can we interpret what those functional links are? And then again, what might this be a useful adjunct to other ways of presenting data? Okay, and so in my last 2 seconds, there's 4 modules in here that have maxillary themes in them, in their expression. So what are these modules? The smallest one has 5 genes. One of them is the clefting gene, LEF1, that we heard Trevor talking about. Not a lot of inspiration here, probably because there's such a small number of genes, it's hard to figure out what the commonality is. But we do know that in a lot of other contexts, using other databases, that these are valid co-expression groups, and so it suggests that there really is something to this. Another group which has now 4 clefting genes in it. This is largely transcription factors, 7 different genes involved in gene expression. A lot of the favorites of those of us who are thinking about craniofacial development, and very suggestive of a network, transcriptional network, regulating facial gene expression. Third network of the 4 in the maxilla or module is heavy in microRNAs, as well as transcription factors, an RNA splicing factor and a transcription factor. Obvious interpretation here is that we have a cohort of microRNAs that are going to be regulating some function yet to be defined. And then there's one more module that's really just way too large to tease apart. It's got almost 300 genes in it. This is your classic hairball, and it's beautiful and it's ugly. Okay, and so the prospect for the future on this is really too large to deal with, but these modules were generated just with our very small subset of data. And if we were to incorporate Yang's data and a lot of the other data out there, I'm very sure these modules, these hairballs, would break out into smaller, more useful modules. And so the vision is something, whoops, yeah, just skip that. So we're starting with this data here, and if we add in so much of this other gene expression data that's out there relevant to the face, along with the data from mutants and other kinds of RNAs, then what we should be able to come up with is a series of modules that describe the genes, the relationships of genes in facial development. And the prospect would then be that, so you have in your GWAS, you've come up with a gene that you don't really understand. It doesn't have a whole lot of annotations associated with it. Well, so you could go and look into the module, what else is in that module, that co-expression module, and really get then some potential insight hypotheses about what your new gene might be doing. Or if you come in and you've got, you're interested in what's going on in the developing taste bud, and you want to know the other genes that are expressed in that time and place in these modules again. So the idea I'm just putting out there is that these kinds of co-expression modules, if we really broadly integrate all the data that we have at FaceBase, would be, I think, really complementary to the tissue and age-specific ways of representing data. And so that's where I want to end this. Thanks.
Okay, thank you, Joan and Trevor. Okay, let's take a couple of questions before we go to break.
Joan, that was really interesting. What aspects of this, what do you expect of this to put on FaceBase, and in what form?
Okay, so it's not really terribly hard to generate these modules. Actually, the hardest part is inputting all the data and co-processing it in a way that you can build the modules appropriately. But yes, my vision would be that these modules would go up on FaceBase
as pictures, or
as gene lists and as these network pictures that I was showing you. So that, and I didn't really explain, but those network representations are really pulling database information about physical interactions, co-expression, tissue, really all the database information that's out there. And so that would be my vision of how these modules would be presented.
Okay, and who else? Somebody over here.
Thanks. That was actually a really interesting talk. I think these are great ways to start analyzing the data that's in FaceBase. One of the things I just wanted to point out is you might consider. I noticed that you used David to do the gene ontology analysis. You probably know that the Go data in there is really out of date. It's from 2009, so you might get additional insights.
So something I didn't actually
reanalyzing with more recent data.
What I didn't actually say is that in the 359 genes, those were all pulled from manual annotations out of the literature because Go is so hopelessly out of date.
Go is out of date. You mean in David?
I'm sorry.
Yeah, David is. Anyway.
Okay. There's two pieces there. David is out of date, and Go is really not geared towards the kinds of questions that we're asking. And so by generating my own annotations, I was really able to see, yes, what I wanted to see.
Yeah, I'd love to talk to you offline about that. I'm one of the PIs at GovGo, so we'd love to hear what we can do to make it better. No, this is great. Criticism's the best thing we get, right? It's a community project, and love to get your annotations in there.
Sonia's over there in the white jacket. She used to work in Larry Hunter's lab and did a lot of stuff on database mining and working on the analyzer and other ways to annotate data. So you might want to chat to Sonia afterwards.
Oh, yes. Okay. One more.
So the data is really interesting. I was sort of struck when you were looking at the different pathways, about how many multiple growth factor signaling pathways you were showing, because you showed some RTK stuff, you showed some BMP, you showed some sonic hedgehog, and so on. And one of the things that I'm thinking is that, first of all, you could manually annotate it like that, because as you were saying, Go is so hopeless, and it's very artificial. Sometimes these classifications that they put in is hopeless. But the other thing that I'm thinking for you and for actually everybody else, it's just a comment, is that it might be useful even on FaceBase, and maybe you've thought about that, is to link all of the genes that you've identified to MGI. Because there's a lot of data which is in MGI, and that would actually, at least on the mouse, which is the mouse genome informatics. And there's also, for people who know how to manipulate it, they also have all the data. They have the databases on the phenotypes. They have the databases on the conditional mutagenesis. And it might be nice for FaceBase to tie in somehow or the other to MGI.
So I'm going to let Melissa deal with tied to MGI.
And that is the subject and title of my talk for tomorrow. So is how do we actually kind of put together some of those pieces so that we can do those kinds of integration.
Okay. And while she's taking the mic over to Sonia, I just want to say to Phil about the signaling. I have a huge chunk of actually manually annotated signaling pathways where separating out the sort of sending things like the ligands and then the receptors, the receiving things. And that there's a really cool, huge thing about mapping those together so that you can actually see the signaling between the tissues. Okay.
Okay, so can you hear me? Actually, I don't know if Trevor told you about both data sets, but when there was the whole prominence data set. Oops. Hello? Testing, Houston. On the data set that Trevor had earlier, which is the one that I was primarily involved in on whole prominence, we did pull in the mammalian phenotypes, and it was nice because you could see the kegs, you could see Go, you could see mammalian phenotype, and I had them as biologists annotate which of those categories were specific for this disease, and you highlight them in blinking red, and having the full list and then the specific ones was really helpful, and very often it would, as expected, pull in abnormal facial topology. So it's something that was done in the earlier data set, and then now Joan has done a great job with the later data set. So we can put them all.
I'd like to respond to Sonia.
Okay, Phil.
No.
It's a 10-minute break now.
I'm also part of Comp 2, and well, I'm not part of Comp 2, but I'm associated
Close.
randomly with Comp 2. And they, I think, have started to do a job now of linking each gene also with a disease, human disease. So that's something else we want to think about, I think, in FaceBase, is whether to annotate the genes in some way that is like, this is a prospective candidate for a particular human syndrome or problem involved in facial development.
Okay, great. Thanks, Trevor and Joan.